Hello everyone, welcome back to the session. So we are discussing about the very short answer questions of in the first year as well as in the second year. So in that line today also we are going to discuss about the very short in the animal diversity one invertebrate phyla. Okay, so in the first year. So the first question will be, what are all the functions of the canal system of sponges? Okay, so you should know the sponges will be coming under the phylum Porifera, and this Porifera phylum has the unique characteristic features of canal system, and the question is asking what are all the functions of canal system of sponges. Okay, the sponges possess a water transport system or the canal system. Okay, so this is an important line. Even though they are asking about the functions, if you give one line of definition, it will be an add-on points. Okay, so the sponges possess a water transport system or the canal system, which means what? The canal system means a water transport system. The water has to be enter into the body and it has to exit out from the body. So all these things have been mediated by the canal system. Okay, now coming to the function, the gathering food. Okay, so gathering food, that is the filter feeder. So food gathering is also one of the function of the canal system. And the exchange of gases. Exchange of gases means the what are all the required gases that has to be taken by the body and it has to exhale out the waste gases. Okay, so that is what an exchange of gases. The respiration is also an important physiological function of the canal system. Okay, and removal of waste. Okay, see, the food is gathering and the respiration, the gases has been inhaling and exhaled by the organisms with the help of the canal system. And also after the food has been digested, the waste materials which is undigested, that has to be eliminated out from the body. So the elimination function, that is the excretion function, is also mediated by the important system called the canal system, that can be seen only in the sponges, not any other phylum. Okay, in the animal kingdom. And also you should know that, and the main important thing will be most of the physiological functions will be done by this canal system. Okay, so this is the proper answer for this question. Now the two chief morphological body forms of cnidarians and their function. See here, so they are asking about the cnidarians. There are two morphological body forms, and that is what the cnidarians body forms and their functions. What is their function? But before that, you should know this cnidarians, they are having this is also called as a coelenterata. Okay, phylum. And they are having the concept of polymorphism. Okay, polymorphism in the sense many forms of body parts. Okay, so that is what the two important morphological body forms will be, polyp and medusa. Okay, so you should know this polyp is sessile, which is sedentary in nature, which can't move from one place to another. It has to be attached with the substratum. Okay, that is what sessile. Okay, and it is cylindrical form in nature. Okay, so the shape of the polyp will be the cylindrical form, and this polyp is the one producing the medusa in the form of asexual reproduction. Okay, we are having different types of asexual reproduction, and the polyp is producing the medusa in the budding type of asexual reproduction. Okay, so these are all an important points. Okay, so just see here, sessile means sedentary. Okay, it is attached with the substratum, can't move from one place to another. And the cylindrical form, the shape of the polyp will be the cylindrical form, and this polyp is the one producing the medusa, that too in the asexual reproduction, specifically budding. Okay, so next, medusa. Medusa is an umbrella shaped. It will be look like this, and this is an umbrella shape and free swimming form. Okay, see, polyp is just opposite to medusa. So polyp is sessile, that it is sedentary, it is fixed in one place, and medusa is free swimming in nature. See, free swimming, and they are producing polypoid forms sexually. Okay, see, they are reproducing sexually, and they are producing polypoid forms, many forms. Okay, So this is what the two chief morphological body forms. So further, this polyp has been occurring in different forms, and this medusa is also occurring in different forms. Okay, so that is why the phylum Cnidaria or Coelenterata, they are having a concept of polymorphism. Okay, poly in the sense many, but we have discussed here only two most important. Further, this polyp is exhibited in different forms and medusa is exhibited in different forms. That is a different question. Okay, but in this question, this answer is sufficient, right? Now the second question, what is metagenesis? Which phylum exhibit it? See here, metagenesis means the another name of this metagenesis is alternation of generation. Okay, see, alternation of generation. Basically, this alternation of generation is seen very clear in plants. Okay, so alternation of generation can be seen clearly in the plants. Okay, see here in plants, the sporophytic phase, sporophytic stage and the gametophytic stage has been alternate in their generation. In simple forms, we can explain that as asexual reproduction followed by the sexual reproduction, as both the type of reproduction seen in the life cycle. So that process is called as an alternation of generation. We can call this alternation of generation as metagenesis. Okay, now this phylum is Cnidaria. Okay, Cnidaria, and the polyp produces medusa by budding. Just now we have studied that polyp in the form of asexual reproduction, that too specifically by budding, they are producing a medusa, right? Now medusa producing a polyp in the form of syngamy, which means fusion of gametes, fusion of male gamete and the female gamete, that is called as a syngamy, and that too, that this type of reproduction is sexual reproduction, as both asexual and sexual reproduction taking place in the life cycle. We can call that as the alternation of generation, that is technically called as the metagenesis, and this concept can be clearly seen in the phylum Cnidaria. Okay, so this is the important question. Okay, now distinguish between amphids and plasmids. Okay, see, what is it? Let us discuss about that. See here, this amphids and phasmids will be seen in the nematodes. Okay, nematodes means phylum Asphelminthus. Okay, nematodes. So this phylum Asphelminthus is also called as nematoda. Ascaris will be the best example for this roundworms. Okay, that is the nematoda. It is also called as Asphelminthus. It is also called as the roundworms, and the best example will be the Ascaris. We have discussed about that. Okay, see, amphids. Amphids means cuticular depressions around the oral region. Okay, around the oral regions, that can be said as the mouth region. Okay, around the oral region we can see the cuticular depressions. Okay, so what exactly this cuticular depressions, why it has to be present around the oral region, means it function as a chemoreceptors, which means they are going to receive some signals. Okay, receive some, sense some chemicals. Okay, some pheromones. Okay, so that is what amphids. Okay, see, phasmids means posterior glandulosensory structures. So those structure which is present in posterior side is phasmids, and in the anterior side, if there is a cuticle depression, it is called as the amphids. Okay, see, posterior glandulosensory structures. Glandulo means, as the name suggests, the glands, definitely it is going to synthesize something. Okay, so that is what, in some glands and the sensory structures which is present in the posterior area, so that is called as a phasmids. So both this amphids and the phasmids are found in the nematodes. Okay, which is Asphelminthus, also called as the roundworms, right? So this is the best, this is the proper difference between the amphids and the phasmids. Okay, now what is metamerism? Okay, see, metamerism means, see, just to see here, consider this as a body of an organism. Okay, see, this separation, this is the segments. Okay, so the segments is also technically called as a metameres. Okay, and this concept of showing this metameres, the concept of showing the segments in its body, that process is called as a metamerism. And this concept of metamerism has been started to arise from the phylum Annelids. Okay, so this is very important thing when it comes to non-core data. All right, see, metamerism means division of body into segments. The segments is technically called as a metameres, and it is separated by a septa. Okay, so this is the body of an organism, and it is divided by dividing the body into a segments. That segments is technically called as a metameres, and this concept of exhibiting the metameres is metamerism, and this can be clearly seen in the annelids. This annelid is also called as the segmented worm. Okay, and the best example for the phylum annelids will be the earthworm. Okay, Nereis, leech, all these having the segments both internally as well as externally. What is the difference of this external segmentation and internal segmentation? Here is a very important difference. See, since the concept of metameric segmentation can be seen in annelids, arthropods, chordates. Okay, see, we are having a segments in annelids, chordates and arthropods. Okay, so none of the other phylums have this concept of segments within it, but among all these things, annelids, arthropods and chordates, when compared to all these things, which a phylum has a proper segmentation is phylum annelids. Okay, see, phylum annelid have the proper metameric segmentation. Why we are saying proper metameric segmentation, improper? What is the difference? See, proper segmentation means we can see the segments externally. Even if we cut open the organisms, we can see the segments internally. If we can see the segments both internally and externally, we can call that particular thing as a proper metameric segmentation. If the segments present only externally and if only internally, they are also having a segments, but it is not a complete, which is not a proper like annelids. So and the arthropods and chordates have only external and only internal. Okay, so this is the difference between all these things. Now coming back to this question, so this metamerism, now you know what is meant by metamerism, and it is very well seen in the annelids, and pseudo metamerism in tapeworms. Now comes the another concept of pseudo metamerisms. Usually the segmentation, see, the pseudo metamerism will be seen in the tapeworm. Tapeworm is coming under the phylum Platyhelminthes. Okay, Platyhelminthes are having the pseudo metamerism, which means the segments, which is called as a proglottid in case of phylum Platyhelminthes, formed at the anterior end. Okay, no true internal segmentation. The internal segmentation will not be seen in the phylum Platyhelminthes. So this is the correct answer for this question. Okay. Now, which arthropod is called as a living fossil? See, living fossil in the sense, the fossil which is present now. Okay, that is name its respiratory organs. Which arthropod is called as a living fossil? So, Limulus, which is a king crab, it is coming under the class Crustacea under the phylum Arthropods. This arthropod is considered as a largest phylum in the animal kingdom, and they are having a class Crustacea, Arachnida and Insecta. And the king crab Limulus is coming under the class Crustacea. This is the one we are calling them as a living fossil. Okay, and the question asking name its respiratory organ. Their respiratory organ is book gills. Okay, see, spiracles. Spiracles is also seen as is also considered as a respiratory organ in case of arthropods, and book lungs is also the respiratory organ present in the organisms under the phylum arthropods, and tracheids. Okay, so tracheae, all these were the respiratory organ, but the book gills is the one present as a respiratory organ in case of king crab, that is the Limulus. Okay, so that is this the answer for this question. Now, what is Aristotle lantern? An example of animal possessing it. Okay, so you should know what is Aristotle lantern. Okay, see, you should know this is the mouth part of some organisms, and you should know this Aristotle lantern will be present in the phylum Echinodermata. Okay, Echinodermata. Echinodermata means in their spiny skinned organisms. So those organisms which are considered as spiny skinned organisms are placed under the phylum Echinodermata. And this Aristotle Lawton, they are having a 5-jawed masticatory apparatus found in the mouth of sea urchin. Sea urchin is coming under the phylum Echinodermata. The example will be Echinus, that is the technical term of the sea urchin. So this is an important question again. Now, the symmetry between juveniles and adults of echinoderms. Okay, see what is the difference between juveniles? Juveniles means smaller ones, younger ones, and adults of the echinoderm. The best thing here will be the symmetry. Okay, so radial symmetry. We are having symmetry, radial symmetry, and bilateral symmetry. Radial symmetry, consider this as an organism. If this organism, if we cut open like this, it will be divided into 2 equal halves. Okay, consider this as an organism. If we cut like this, we can come up with 2 morphologically equal halves. If we cut like this, again we can come up with 2 morphologically equal halves. If we cut like this, again we can come up with the 2 morphologically similar halves. Okay, so that is why if an organism at any plane, if we cut the organism at any plane, we can come up with 2 morphologically equal halves. So that type of symmetry is called as a radial symmetry. Okay. Now, juveniles means consider this as an organism, and this is an eye, okay, and this is a nose, okay, and this is a nose, and this is the mouth. If we cut at this plane, here one eye, here one eye, here half of the nose, here half of the nose, here half of the mouth, and here half of the mouth. Okay, only at one plane, if we cut a particular organism, we can come up with 2 morphologically equal halves. So that is what the difference between radial and bilateral symmetry. And the radial symmetry, which is present in the echinoderms, we can call them as a pentamerous radial symmetry. Why? Okay, just to see here, this is a starfish. Okay, starfish is coming under the echinoderms. Okay, we can cut the I'm sorry, 1 minute. See, this is the starfish. Okay, we can cut an organism like this. Again, here a morphologically equal halves, here a morphologically equal halves. We can cut like this. Okay, we can cut like this. Here 2 equal halves, here also a similar halves. Okay, so that is why, as it has 5 arms, we are saying that as a pentamerous radial symmetry. Okay, so this is what the difference between juveniles as well as the adults of the echinoderms with respect to the symmetry. Okay, the question is asking with respect to the symmetry, right? Now, what are all the blood glands in perithema? Okay, see, blood glands in perithema. So, but before that, you should know what is perithema. Perithema is nothing but an earthworm. Okay, see, earthworm which is coming under the phylum Annelids. So, as we have mentioned before, the annelids have the unique character, which means not unit. It is a special characters which has a metameric segmentation within it. And the question are asking about the blood glands with respect to the perithema. Okay, see here, it is located in 4th, 5th and 6th segments. 6th segments of the earthworm, they are producing blood cells. Okay, those organs which is present in each segments, they are responsible for the production of this blood cells and the hemoglobin which is dissolved in the plasma. And you should know blood cells are phagocytic in nature. Phagocytic means what? They are considered as the they are attacking cells. So if any enemy enter into the, if any microorganism enter into the body of the perithema, these blood cells have the capacity to engulf them, engulf the microorganism and digest them. Okay, so that is what a phagocytic in nature. So this is what with respect to the blood glands in the perithema. Okay, so now what are spermathecae on the body of perithema? Okay, see here in perithema, what is mean by the spermathecae? Okay, see here, from the segment 6 to 9, from the 6th segment to 9th segment, they are having the 4 pairs of spermathecae. Okay, 4 pairs, 6, 7, 8, 9. Okay, so 4 pairs, which means what? Totally 8 spermatheca. Okay, because they are asking about 4 pairs. They are saying that 4 pairs, which means 8 spermatheca, which is located in the 6th segment to 9th segment, which means what? Each segment have 2 spermatheca. Okay. Why it has to possess the spermatheca in these segments? So what is their function? They receive and store the sperms during copulation. Okay, copulation in the sense the fusion of male and the female. Okay, so that is what the spermatheca, right? So this is very important question again. Now, so that's all about the short answer question in the animal diversity 1 in the phylum non-chordata. Now comes the animal diversity 2 in the phylum chordates. Again, this belongs to the first year, and the first question will be, name the type of caudal fin and scales that present in a shark, cutla respectively. See here, type of caudal fin. Okay, so the caudal fin and the caudal fin as well as the scales. Okay, scales which is present in the shark and the cutla. See here, caudal fin means the fin, and the fin which is present in the tail region is called as a caudal fin. Okay, so in case of shark and the cutla, the caudal fin will be heterocercal and here it will be homocercal. The word hetero means different, homo means same, and here it will be different, here it will be the same caudal fin. And the scales which is present in the shark and the cutla will be the placoid scales will be seen in the shark, and the ctenoid or cycloid scales will be present in the cutla. Okay, so this is also a important question. And how do you justify the statement heart in fishes is a branchial heart? See here, what are they saying? See, heart in fishes is a branchial heart. Why we are saying heart in fishes is called as a branchial heart? See, here is an answer. Because it supplies a blood only to the gills. So as it supplies, so the branchial heart, the fish's heart is called as a branchial heart. Why? Because this heart which is present in the fish, which is supplying blood only to the gills, not any other else. Okay, so that is why the fish heart is called as a branchial heart. This is an exact reason. Okay, now what are claspers? Which group of fishes possesses them? Okay, claspers. So you should know claspers is somewhat related to fertilization. Okay, so fertilization specifically, this is the structure which is present in the male reproductive organ. This belongs to male reproductive structure. Okay, so claspers, they are the structure on the pelvic fins of the male chondrichthyes. See, male chondrichthyes. Chondrichthyes means we are having two different types of fish. One is cartilaginous fish and another one is bony fishes. And this chondrichthyes is the means it is cartilaginous fish. Okay, so in the male cartilaginous fish, in the pelvic fins, they are having a structure called the claspers. Okay, and this claspers which is present in the example for the chondrichthyes will be the shark, and it is used for internal fertilization. So what is meant by fertilization? Fusion of male and female gametes. Okay, and what is meant by external and internal fertilization? If the fertilization happens outside the body of the female, it is called as a external fertilization. If it happens inside the body of the female, we can call them as an internal fertilization. Okay, so now this is for the internal fertilization, right? So all these were the important keywords. Now, how does the heart of an amphibian differ from that of a reptile? See, we are comparing the amphibian and the reptile, okay, and heart. And you should know that, see, fishes, amphibians, reptiles, and aves, mammals, okay. So they are having two chambers of heart. Reptiles are having three chambers of heart. Aves and mammals are having 4 chambers of heart. See here, amphibian heart and reptile heart. We know that amphibians are having 3 chambered heart, and reptile heart will be the 4 chambered heart except the crocodile. See, actually it is a 3 chambered heart. Again 3 chambered heart except crocodile. All these are having 3 chambered heart except crocodile. Crocodiles are having 4 chambered. Okay, now see, the sinus venosus is present in the amphibian's heart. Again the sinus venosus is present in the reptile's heart. Conus arteriosus is present in the amphibian heart, and the conus arteriosus will be absent in the reptile heart. Okay, so these are all the important important difference between the amphibian heart and the reptile heart. Just see here. See, both are the 3-chambered heart except crocodiles. Crocodiles are having 4-chambered heart, and the sinus venosus is present in both amphibians heart as well as the reptile heart. The conus arteriosus is present only in the amphibian heart but not in the reptile heart. So these are all an important difference. Now, how do you distinguish between a male frog and a female frog? And this is very important here. See, male frog, they has ocal sacs, copulatory pad on the first digit of each forelimb. See, they are having forelimb. Okay, in each forelimb, on the first digit of each forelimb, they are having a ocal sacs to make a sound, and copulatory pad for the copulation, for the fusion with the female frog. Okay, see, female frog, they lack both ocal sacs as well as the copulatory pad, because all these things are need only for the male frog, they don't need for the female frog. So this is the major difference between the male and the female frog. Now, the distinguish between milt and the spawn. See, milt means mass of sperms released by the male frog. This is a technical word, okay. The male frog is releasing a sperm, okay, and that sperm, it is not only one, it is a mass of sperm, okay, and the mass of sperm released by the male frog is called as a milt. Okay, and the female frog is releasing an egg, and that will be in the mass. So that mass of the egg will be in a mass of egg is called as a spawn. Okay, so this is the word somewhat related to the gametes, right? So the next question. Name 2 poisonous and non-poisonous snakes found in the South India. Okay, see, poisonous, as it is very famous, Naja. Okay, cobra. Okay, cobra is considered as very poisonous, and Bangaras, that is a crate. Okay, crate is considered as a poisonous. Non-poisonous, Tyus, that is a rat snake, and Tropidonotus. Okay, pond snake. So all these snakes were non-poisonous in nature. They can bite, but it is not a poisonous snake, right? Now, name the 4 extra embryonic membranes. Okay, see here. There is an embryo, and this embryo is surrounded by a 4 embryonic membrane, and that membrane will be amnion, which has an amniotic fluid within it, allantois, chorion, and yolk sac. All these were the 4 extra embryonic membranes present in the female, which means in the embryo within the female body, right? Now, distinguish between altricial and precocial hatchling. See, altricial, precocial, okay, an important word. See here, this altricial hatchlings means, again, hatchling means hatching, okay. See, it's seen in the flying bird, okay, the seen in the flying bird, incapable of movement after hatching, okay. See, this type of hatchlings, it is seen in the flying birds, and it is incapable of movement after hatching. Immediately after hatching, they can't has a capacity to move immediately, okay. And the precocial hatchling means it's seen in the flightless birds, okay, those birds which can't fly. So that is what a flightless birds, capable of movement soon after hatchling, okay. It can move soon after hatchling. That is what precocial hatchling and altricial hatchling, okay. Important question. Now, so we are going to discuss about the very shorts in the body fluids and circulation in the second year, okay. So first question will be, sinoatrial node is called as a pacemaker of our heart, isn't it? Of course, we know that the famous statement is SA node. There is a sinoatrial node which is present in the right atrium. Okay, a right atrium, in the upper side of the right atrium. Okay, so the right sinoatrial node which initiates and regulates the heartbeat by generating regular electrical impulses. Okay, they are the one initiating as well as they are the one regulating the heartbeat by generating an electrical impulse. So this is why we are saying this SA node as the pacemaker of our heart. Okay, it sets the pace of the heart contractions, hence called the pacemaker. It sets the pace of heart contractions, which means it has to contract now, it has to relax now. The time gap between contraction and relaxation, and the rhythmic contraction and relaxation, that is what the pace of the heart. Okay, so this the sinoatrial node is the one setting the pace of the heart contraction. So that is why we are calling this SA node is called as a pacemaker of our heart. Okay, now The second question will be, name the valves that guard the left and the right atrioventricular apertures in man. Okay, left atrioventricular wall and the right atrioventricular wall. They are asking about what are all the walls which is present in the heart. Just look at the picture here. So this is considered as a heart. And okay, so this is what a heart, and here we are having one chamber, here one chamber, and here one chamber, and here one chamber, and we are having three walls here, and we are having two walls here. Okay, so this is an atrium, and this is ventricle. Okay, this is ventricle. So the septum which is separating this atrium and the ventricle, it is called as the atrioventricular apertures. And you should know this is the right side and this is the left side. Okay, this is the right side and this is the left side. Okay, so those aperture which is present in the right side is called as a right atrioventricular aperture, left atrioventricular aperture. Okay, and the walls which is present in the left atrio aperture, which is have just two openings, and that is called as a bicuspid wall. And this bicuspid valve is also called as a mitral valve. And the valve which is present in the right atrioventricular valve is tricuspid valve. So this is a important thing. Okay. Okay. So this is the answer. Next step. Now comes the another very short question answer from the excretory products and their elimination again in the second year. Okay, so the first question will be, name the blood vessels that enter and exit the kidney. Okay, blood vessels that enter and exit the kidney, that is the renal artery and the renal vein. Okay, see, we are having two blood vessels that enter and exit the kidney. Okay, one blood is carry the blood and it is taking to the kidney, and one is carrying the blood and it is taking away from the kidney. Okay, see, that is what a renal artery and renal vein. Always the word renal refers to the organ kidney. Okay, the renal artery means it entering the kidney, and the renal vein means exit from the kidney. Okay, it is coming out, it is entering into. Okay, so that is what the question. Now, what are all the columns of Berton? Okay, columns of Berton are extensions of renal cortex between renal prummets in the medulla. They are the one providing support and passage for blood vessels. See here, there is an important structure which is present in the kidney, that is the columns of Berton. So they were the extensions of renal cortex. Renal cortex is nothing but the outer region of the kidney is called as a cortex, and the inner region is called as a medulla. Okay, see here, there is the extension from the outer region, that is nothing but the renal cortex. The extension from the renal cortex is called as a columns of Berton, and that is between the renal prummets in the medullary region. So the inner region is called as a medulla. In the inner region we are having a prummet, and this columns of Berton will be present between the prummets in the medulla. Okay, so this is very important structure. And what is the function of this columns of Berton means? They are providing the support as well as a passage for the blood vessels. Okay, now the next question will be glomerular filtration. See, glomerular filtration is a process by which the blood is filtered under the pressure through the glomerulus into the Bowman's capsule, forming the glomerular filtrate. See, actually glomerulus is the structure which is present in the Bowman's cup, which means in the kidney. Okay, so there is a structure which has the glomerulus. Okay, so which is present in the Bowman's cup or Bowman's capsule. So the filtration done in the glomerulus is called as a glomerular filtration. How it is exactly happen? It is a process. Okay, blood is filtered under the pressure. Okay, that's what a blood pressure. The blood is filtered under the pressure through which structure? Through the glomerulus, which is present in the Bowman's capsule. This is the one forming the glomerular filtrate, which means the filtration of the blood occurs in the glomerulus is called as a glomerular filtration. Okay. So the distinguish between the juxtaglomerular cells and the macula densa. Just to see here, the juxtaglomerular cells means it is a modified smooth muscle cells. Okay, modified smooth muscle cells in the wall of efferent arteriole secrete renin. Very important. See, we are having an afferent arteriole and efferent arteriole. Just now we have studied, one is taking in, in the kidney, one arteriole which is entering into the kidney, one is taking away from the kidney. If it is entering, we are calling them, the arteriole enter into the kidney, we are calling them as an afferent arteriole. If the arteriole coming out, that is called as an efferent arteriole. Okay, but here, it is a juxtaglomerular cells, it is a smooth muscle cells which is present in the wall of afferent arteriole which is entering into the kidney. Okay, and these are all the cells are responsible for secreting a renin. Okay, we are having two renins. One is renin and rennin. Okay, and this is with respect to the kidney, renin. Okay, so the macula densa is the one. They are the specialized epithelial cells which is present in the distal convoluted tubule, that is the DCT. So in the nephrons we are having the glomerulus Bowman's cup, PCT, proximal convoluted tubule, Henle's loop, and after that distal convoluted tubule, and after that collecting duct will be there. And in the distal convoluted tubule, we are having a specialized epithelial cells that is called as a macula densa. Okay, so what is the point of having this macula densa in DCT, that is a distal convoluted tubule? They are the ones sensing the sodium concentration and regulate the renin release. Okay, so the renin release by the juxtaglomerular cells, which was decided by the macula densa by sensing the sodium concentration in the distal convoluted tubule. This is the correct answer for this question. Now, the last question will be difference between cortical and juxtamedullary nephron. See, what do we have discussed before? In the kidney, we are having the outer region called the cortex and the inner region is called as a medulla. Isn't it? Now, so the nephrons is the structural and functional unit of the kidney. If the kidney which is present in the outer region, that is in the cortical region, we are calling them as a cortical nephrons, and if it is present in the medullary region, we are calling them as the medullary nephrons. Okay, see here, cortical nephrons, there is a cortical nephrons is present in the renal cortex, and juxtamedullary nephrons will be near the cortex medulla junction. Loop of Henle will be very short, and here in the juxtamedullary nephrons it will be long, extends deep into the medulla. Okay, and percentage in kidney, it will be approximately 85% of cortical nephrons will be there, and only approximately 15% of juxtamedullary nephrons will be there in the kidney. Okay, and what will be the significance in urine? Significance in urine synthesis with respect to the cortical nephrons will be, it is less significant when compared to the medulla. Okay, it is very crucial for the concentrating urine. Why? Because of the presence of the Henle's loop. Okay, present in the juxtamedullary nephrons. Okay, so that's all about today's class. We'll meet you in the next class with another chapter, short answer question. Thank you so much.